going to move right on to Dr. Susan Bookheimer, who's going to introduce advances in brain imaging that really get to connect with what we call individual differences in autism.
Okay, great. Thank you very much. Very pleased to be here. So you've heard a little bit about some of the individual differences that we see in autism, and I like to coin Dan Geschman's phrase, if you've seen one kid with autism, you've seen one kid with autism. So there are many individual differences that we can look at, and what I'm going to be talking about today is what do we know about differences in the brain, particularly structural and mostly functional brain imaging, and how do they relate to some of these individual differences, and can that help us to understand better what are some of the underlying causes of the variation that we see in ASD. I have no disclosures, but if you want to pay me later, you're welcome. Okay, so as we said, there is substantial individual variation among individuals with ASD, and I think it's interesting that some of this is appreciated in the new DSM. So you can now choose from a family of potential phenotypes in addition to the two core deficits. And so I'll be addressing the question, do these individual differences reflect differences in brain structure and function? I hope at the end I'll have just a minute. I'm just going to talk about one gene, but I think that there's a broader question. Do these brain differences relate to variation in genetics and genetic risk for autism, which we think is probably the case? So some of the phenotypic variations I'm going to talk about: degree of repetitive behaviors, severity of the social symptoms, social communication symptoms, the presence of anxiety or sensory over-responsiveness, and then more generally network connectivity and global deficits in autism, as well as the genetic background. So let's start with repetitive behaviors. As you know, repetitive behaviors can take on many forms, and they can be very mild, and they can be very, very severely disabling. There have been a couple of studies looking at brain development, and particularly structural brain development, and its variation in repetitive behaviors. In particular, I want to point to this longitudinal study of structural development looking over a 2.5-year follow-up period in preschoolers, where they're finding the primary differences in the striatum, particularly the caudate nucleus, associated with motor sequence performance and OCD-like behaviors that we see in autism. And they examined the rate of growth in the caudate. So for those of you who are not fully versed in the striatum and the caudate, I just have a couple of slides just showing you background. This is important because I think the striatum is one of the structures in various parts of it that are highly implicated in autism, and I'll show you that as we go along. So this is, if I can get my mouse, there we go. So this is the whole brain. The striatum, the basal ganglia, is a set of very, very deep structures in the brain, and this is a close-up of this. So you can see there's the caudate, putamen, globus pallidus, thalamus, and the caudate has this big old long tail and ends in the amygdala. So we don't usually get to see it like this. We usually get to see it like this. Here it is in the actual brain. This is an axial view, so that's cut through this way, and the other one is a coronal view, so cut through this way. And so here is the caudate, putamen, globus pallidus, and then from a coronal view, caudate, putamen, globus pallidus, and down here is the ventral part of it, which we'll call the ventral striatum. So this would be more dorsal striatum, and this would be more ventral striatum. So the striatum is very important in many things that are related to other skills and other abilities and disabilities, but also related to autism. It's involved in motor learning, motor sequence learning, skill learning. It's involved in implicit learning, so learning without consciousness, learning by repetition. And it's also important in reward-driven learning. It is implicated in decision-making based on outcomes. So basically making a decision based on your implicit assumptions about whether you're going to have a positive or a negative outcome. It's implicated in movement disorders, so Parkinson's disease is affected in this area of the brain, Huntington's disease, a lot of other movement disorders, but it's also been implicated in obsessive compulsive disorder, in addictions, and in ADHD. So this is a very important area of the brain with a lot of different substructures with functions that are relevant to many of the problems that we see in autism. So just from a structural perspective, this one study looked at two parts, the caudate and the putamen here, and related the amount of gray matter volume to the, and change in gray matter volume over this 2.5-year period to the repetitive behavior aspect of the ADIR, the insistence on sameness. Typically what happens in development, by the way, is that we start out with a whole bunch of neurons and our gray matter starts to decrease a little bit as we prune and refine what neurons we want to keep and what neurons we want to prune. But here what we saw is this increase in trajectory of brain growth, an accelerated brain growth, so to speak, that was correlated with the worst symptoms, with increased symptoms in repetitive behaviors. So it suggests that there's something specific to these areas of the brain that is structurally related to repetitive behaviors. Most of what we do here is functional imaging, so I'm going to talk to you a little bit about fMRI. I think all of you have heard enough about fMRI that I don't have to give an introduction to it, but basically we're using MRI scans to look at how the brain is actually working while we perform some relevant task. And this is a task, a very commonly used task called an incentive delay task. There are two versions in this particular experiment, a monetary incentive delay and a social incentive delay. But the basic idea is that you get a cue that tells you what the outcome is going to be, whether you're going to get a small reward or a big reward. And in one condition, it's a money reward, you get, and in another condition, it's a social reward. And what you do basically is you press a button. You see a target, this square trigger here in the center. And when you see that, you press a button as fast as you possibly can. And if you press it fast enough, then you'll get the good outcome. So the faster you press it, the better your outcome. In the monetary incentive delay task, which is this one, you see a cue that says, if you see this, you're not going to get a reward. If you see this, you're going to get a small reward. But if you see this, you're going to get a big reward. Then you get the trigger, you press the button as fast as you possibly can. And then you have a feedback stage where you see either nothing, a small, or a large. And it's indicated, you can't really see very well, but it's indicated out on how big your reward is. So that's a monetary reward task. And then this is the social reward task. So you either see nothing, which happens to be a blurred face, and then a small reward is just like a little tiny, almost neutral, but tiny bit of a grin there. And then a larger reward is a big old fat grin. You may not think that that's very important, but it turns out faces are extremely rewarding, and I'm going to actually talk about that in a second. What they found in this study is that there is a group by reward interaction. The top is still not really coming out over there. Is there any way we can make the picture smaller? Just compressed more, so because we're missing both the top and the bottom. Top and the bottom. I'll just go ahead and talk about it a little bit while we're trying to see if, I think it must be in the formatting or something. That's what happens when you use somebody else's computer. Why don't you look at the displays and see if you can change the width of the display. In this study, that won't work. Thank you very much. There's a group by reward type interaction, and in particular, the caudate, this part of the striatum, is decreased in its activity in response to social incentives. And there's this cross correlation so that negative social task activation correlates with degree of repetitive behavior severity. And you can see that in the graph over here. So this is the monetary reward in the ASDs, and then, let me get my mouse, there we go. Monetary rewards and social rewards in ASD, and these are the same in the control children. As you can see, there are very, very big differences in how this part of the brain is reacting in response to rewards. And in particular, in response to social rewards, not only does this area of the brain not activate, it actually deactivates. It goes down towards social rewards. And what's important is that the degree to which this decrease takes place relates to the degree of severity of the repetitive behaviors. So there is an abnormal structure in this area, but there's also tremendous abnormal function that appears highly specific to the social aspect of the stimulus, anticipating a social reward, and which is clearly related to behavioral, to the repetitive and restrictive behaviors. So to summarize just this part of the talk, the severity of repetitive behaviors in ASD is related to dysfunction in the structure of the basal ganglia, which is a collection of regions that are important for complex motor behaviors and outcomes or reward-based or decision-making. Abnormal growth of these regions is related to severity, and abnormal function of these regions is related to severity just of this repetitive behaviors phenotype. So what about other important phenotypes? Social impairment, the degree severity of social impairment is obviously a very important phenotype and can vary quite a bit in autism, although everybody has it to some degree. And so here I'm going to talk about a few different kinds of studies, one focusing on reward processing, one focusing on mirror neuron regions, and then I'm going to talk about the development of independent brain networks, I'll explain that a little bit later. Let's talk about social motivation and social rewards. Ted already mentioned the social motivation hypothesis of autism, and it's a very important hypothesis that has a very strong grounding in the neurobiology of autism. So social rewards, we believe, are primary reinforcers. So in the brain responds to, does not have to learn to respond to social reinforcers. It's a primary thing. Neonates orient preferentially to smiling faces. There's reduced orientation to faces, as Ted has already talked about early in autism. It's one of the earliest features that is highly reliable. And so there's a general theory that reward processing deficits could reduce social motivation. If these social reward systems are not well in place, then that would tend to make one not want to orient towards social stimuli, and then that could set off a cascade of events of abnormal learning and abnormal social experience, which sends you further down that road of autism. There's a brain region called the ventral striatum that's very important in rewards, and this region turns out to be important for every kind of reward you can imagine. So it's important for social rewards, it reacts for monetary rewards. It is active in drug addicts who see drug cues. It's active in gamblers who are given a high stakes gambling choice. And it turns out it's also active for if you successfully exact revenge on somebody who's hurt you. So anything that could be conceived of as rewarding, this area is involved as a general reward system. So there are a variety of ways of studying it, but this is an fMRI task that one of our former graduate students, Ashley Scott Van Zeelen, did along with Morella DePreto, my close colleague here at UCLA. Again, using fMRI in a implicit learning reward-based paradigm. So implicit learning is one of the skills that's important in the striatum, and of course reward is also important in the ventral part of the striatum. So in this task, there are a couple of different kinds of conditions, and let me walk you through that. The subject sees a stimulus like this psychedelic design here, and they're told that a number goes with it, 1 or 2. They have to press a number 1 or 2 to see if it goes there, and if they're right, they get a monetary feedback sign, and if they're wrong, they get the X's crossed out. And there's a control condition where they do see the same kinds of things, but they're just told correct or incorrect, and there's no reward quality to it. Now, unbeknownst to the subject, there's not a perfect relationship between the stimulus and the number outcome 1 or 2. It's only probabilistic. About 66% of the time there's a relationship, and the rest of the time there isn't. So you can't really memorize these, but if you practice it again and again, you slowly start to learn these associations. You actually learn them without awareness. So this is the monetary reward condition. And then the social reward condition is very similar, except instead of getting money, you see the smiling face of one of our pretty former graduate students, and if you get it wrong, then you get the pouty face. So we looked in this study at the learning rates for social and non-social stimuli in children with ASD and in typically developing kids. The first thing that's very obvious and we clearly saw was in that area of the brain. Again, here's the striatum, caudate putamen, and this is the ventral striatum, that reward area. So when we look across all reward types, there's a very, very robust response in the reward center in typically developing children. And as you can see, it didn't quite reach significance in kids with autism, and there's a big difference between groups. So it does suggest, at least to some extent, that the reward processing system is not working as well as it should, in the way that it should. Interestingly, this also relates to learning. So as we know, reward feedback increases our learning rate. And if we get rewards, we learn more quickly. And we see that learning curve in the typically developing kids in pink and red, where pink is the social reward and red is the monetary reward. And you'll even notice that social reward learning is faster than monetary reward learning, which might seem a little funny. It's just a picture of a smiling face, but again, that's a primary reinforcer. And notice the learning curves in the kids with autism. They just don't seem to be learning, at least implicitly, on the basis of that feedback, of that rewarded feedback. But more to the point of looking at individual variation, when we look at the activity in the reward center, and of course we can't do this so much in the kids with autism because they don't have enough activity here, but when we look in the typically developing kids and look at how much activity there is in the brain, that correlates with the amount of social responsiveness they show on the social responsiveness scale. And the correlation is that the more response we get in this reward center, the better our social skills are. The less response we get in that reward center, the worse the social skills are. And so it suggests that the brain's ability to respond in this area of the brain, in this reward system, is important for helping us develop social skills. Interestingly, most of the reward studies in autism have used positive rewards as outcomes. But this is a recent study that was done by another group, Damiano et al. And instead of looking at positive rewards, they looked at negative reinforcement, negative outcomes. And so, again, they used the same kind of monetary incentive delay task where you see a stimulus, you have to make a response real quickly, and if you got it right, you didn't get anything. And if you weren't fast enough, then you lost money. And then in the social conditions, same thing. If you got it right, you got a neutral face. If you got it right, you got a neutral face. If you got it wrong, you got the pouty face. You can't see it very well here, but it's a pouty face. And then in the non-incentive trials, which is a different cue, it didn't matter what you did. Whether you hit or missed didn't make a difference. You always got a negative outcome. And so this is a little bit different because this is learning to avoid a negative outcome. So it's a different kind of reward processing. Nevertheless, they saw very important differences again in both the dorsal striatum and the ventral striatum, and it dissociated whether it's a monetary or a social outcome. The typically developing children had more activity in the caudate, in that same part of the basal ganglia, when they were successfully avoiding this monetary loss compared to kids with ASD. And likewise, in the more ventral parts of the striatum that you see over here, in avoiding seeing a sad face, again, the typically developing kids show more activity in this reward center when they successfully avoided a negative outcome compared with the children with ASD. So it suggests that it's not just that this area is responding badly to positive rewards, but it's also not responding well to avoidance of negative outcomes, and particularly negative social outcomes. The children simply weren't responding as well to those negative social outcomes. But again, to our point of looking at individual variation, the severity of the social symptoms was related to the amount of activity in the brain, such that the greater activity in the caudate, in this part of the striatum, the better the social responsiveness scale was. And so those people who had less activity in this area also had poorer social outcomes. And that's true whether you had ASD or were typically developing. So this is an area that's very important, it appears, in developing that social skills, and it's in line with the social motivation hypothesis. I also wanted to mention this other network of brain areas, the mirror neuron system that my colleague Marilla DiPreto has studied extensively. These are a series of areas in the brain, the inferior frontal cortex, this part in the inferior parietal lobule, and in the posterior superior temporal sulcus, as well as some subcortical regions. These are areas that are important in, they're areas that are active when both an individual performs a meaningful motor behavior and when they observe somebody else doing it. And the idea is that this is, first of all, essential to imitation, but it's also essential to understanding what somebody else is doing by internalizing it yourself. It relates also to the limbic system, and it appears to be related to empathy. So the better the system works, the more it seems that you can resonate with another person by understanding what they're internalizing, what they're feeling and thinking. So this is a system that helps us to understand the intentions of others by effectively mirroring what they're doing as if we're doing it ourselves. So in this seminal study from Nature Neuroscience, Rella DiPreto, who is pictured here, showed pictures of different emotions to children with autism and typically developing kids. And the children either had to just observe the emotion or imitate the emotional expression. We actually had them videotaped on the outside to look at whether they were able to make the emotional expressions, and they were rated by blind raters, including myself, and we could not tell based on their ability to actually make the emotional expression. So they could do the task, but what the brain did was very, very different. And I'll just try to call this out a little bit. On the top row is typically developing kids, and some of these areas are visual areas because they're seeing faces, motor areas because they're moving their face in response. But then they're also showing this activity in the inferior frontal gyrus, which is not essential for imitating, but it is one of those mirror areas. And so while they're imitating the expression, they're also mirroring or internalizing what the other person is doing and thinking. When we look at the ASD children, they've got the same activity in the visual areas, they've got the same activity in the motor areas, so they're doing the task, but they don't mirror what's happening in the other person. They don't have that same internalization, and these are the differences between groups. So the question again is, how does this relate to severity of social symptoms? And it turns out that particularly in that mirroring area in the frontal cortex, it's related to social functioning on both the ADOS and the ADI, particularly in the social symptom severity aspects of those scales. So in other words, the more activity in this area, the better social functioning. The less activity in this mirroring area, the poorer the social outcome, the poorer the social communication. So again, it suggests that some of the variation that we see in the severity of these social communication, social interaction systems relates to differences in how the brain itself is functioning. So in summary, the degree of social communication and responsiveness to regions in the ventral striatum as well as in the inferior frontal cortex are related to the severity of social symptoms. And it implicates this frontostriatal circuitry in autism, and in particular in the social symptoms of autism. Here's an area that has not been studied very much, and I think that the reason why is because it hasn't been in the DSM until the DSM-5, and yet it's a major problem in children with autism, sensory over-responsiveness. About 55 to 70% of children with ASD experience sensory over-responsiveness, or SOR, which is an extreme reaction or avoidance of sensory stimuli. And it can be any kind of sensory stimuli, loud noises, visually stimulating environments, sudden touch, and uncomfortable textures. And I think uncomfortable textures is the thing that most of us see very, very frequently. The kids don't want to wear the sweaters with the itchy fabrics or everything. And sometimes we'll only wear the same article of clothing because it's comfortable. So one of our recent graduate students, Shula Green, did a pair of studies, and I'm going to show you both of them, on presenting children with annoying visual stimulation and annoying noises. And so here, they would either hear like this horrible white noise sound, like telephones, the ringing, a horrible white noise sound, or this colorful flashing checkerboard that moved very quickly and it's kind of jolting, or both of them at the same time while they're being scanned. This is a bit of a complicated slide with a lot of graphs, but I think that you can all see they say about the same thing. Here's what is really interesting. So this is looking at variations within individuals with autism and typically developing kids along a dimension of sensory over-responsiveness. So we have a series of sensory over-responsiveness questionnaires. And what we found was that the severity of sensory responsiveness was correlated with hyperactivity, not hypo, but hyperactivity in certain areas of the brain. In particular, it was correlated with overactivity right in the sensory cortices. So visual cortex was hyperactive, auditory cortex was hyperactive, the thalamus was hyperactive. The thalamus is an important region in the brain because it gates sensory stimuli. That is, you can, signals from the frontal lobe can go down through the thalamus to your primary sensory cortices and narrow down how you experience that information. So it's a gating mechanism. And all of those areas show this hyperactivity in response to these annoying stimuli. Also, the amygdala, our primary emotional response system, showed hyperactivity among children with SOR. So she followed this up with a second experiment, and this one, she added the annoying touch. She actually went to a thrift shop and got one of those old itchy sweaters from the 50s and got a piece of that fabric and then rubbed it on the child's hand during the scan. And then the other stimulus was loud environmental noises like cars and honking horns and things like that. So a more environmentally valid stimulation. But actually what she found was that the biggest effect was when children experienced both at the same time. And this harkens back to what Ted talked about this morning, alluded to this sensory integration. When they experienced both annoying stimuli at the same time, there was again this hyperactivity in this case the auditory cortex, hyperactivity in the touch areas, the somatosensory cortex, and hyperactivity in the amygdala. And that activity correlated with severity of SOR symptoms, even within the autism sample. So what this tells us is that variations in the degree of sensory overresponsiveness that we see in kids with autism is associated with a hyperactive response in the brain in these sensory areas, and in the emotional areas it's probably as a result of essentially being out of control of all of the sensory information. One of the interesting things we can do in brain imaging is try to look a little bit more at the cause of these hyperactivities. And so what Shula did in her most recent paper was these stimuli were presented several times across an experiment. And so what she did was she looked at the responsiveness over time and to see how that changed. What was interesting is that in the very beginning of the trials, the kids with sensory overresponsiveness and kids with ASD had the same amount of activity in the brain. But what happens is that over time, typically developing children habituate to that response. But the children with ASD, and in particular these are kids with high levels of SOR, did not habituate. In the emotional areas, right and left amygdala, and in somatosensory cortex, they did a little bit more in the auditory cortex, but it's a much slower rate of habituation. So again, it's not so much that the brain itself is hyperactive, but the mechanisms that are involved in habituation appear to be what is actually what's causing the problem. So in summary of this section, within children who have ASD, sensory overresponsivity is associated with increased brain activity in these sensory areas, in these sensory gating areas, in emotion processing and memory centers, and it's especially strong when there's simultaneous stimuli. It appears to be due to a slower rate of habituation, and I would point out that this is very important when you consider what kind of treatments you would give to kids who have high anxiety and high sensory arousing, because what is our major treatment of anxiety? It's exposure with the assumption of habituation. Well, if you have a group of children who are not habituating, then that exposure may not only not help, but it may actually make the situation worse. This is an example of why we think it's so important to understand the brain mechanisms that underlie these disorders, because it can tell us fundamentally what is going on and change your idea of how you might approach treatment. I wanted to mention maturation of brain networks and how that differs across individuals as well. And this harkens back to some of the things that Shefali talked to you about brain connectivity being impaired in autism. We think developmentally the connections are not formed well, and I'm going to show you some examples of that. During development, one of the key features that happens in development is our brain starts to divide up into functional modules that are highly specialized and segregated from each other. So we end up with a language system, which is separate from our visual system, separate from our auditory system, separate from our attention system, separate from our self-regulation systems. These systems become increasingly segregated as we develop. And we believe that brain maturity is related to autism symptom severity in a more global way. So this is a network diagram of how we look at network connectivity in terms of its modularity and its segregation of networks. So this is from a study of normal individuals. These are normal children, and these are normal adults. And these are two different networks in the brain. This is a dorsal attention network, and this is a ventral attention network. And what you can see is in adults, these two networks are highly segregated from each other. The different brain areas within the network are all connected together, and the brain regions in this network are all connected together very nicely, but they don't connect much with each other. They're segregated, and that's part of a mature brain. In younger children, they still have a lot of connections within these networks, but they also have some cross connections. So one of the important things that happens in development is these networks start to segregate and become more and more independent. They become uncorrelated with each other, and each starts being able to connect very nicely with itself. This is a study that was done recently by a different group, Young et al., that showed that connectivity, I just wrote that wrong. I said lower connectivity. It's actually higher connectivity between the executive and face processing networks predicts, I was right. Lower connectivity predicts reduced autism symptom severity. The more independent these two networks are, the better the social processing. So these are two networks that we think are important in autism. This is a face processing network, which we know is extremely important. Face processing is a critical skill that is impaired in autism. And the other one is the executive control network. And they looked at the correlation between these two networks, which should be segregated, and found that the degree of correlation related to the severity, the global severity of autism. So overall, the message is that the extent to which these networks have matured and formed separate networks relates to better symptom outcome, and the extent to which the networks have not segregated and they're still cross-talking too much suggests more global symptom severity in autism. And this was also found in a different study altogether, looking at different networks, the theory of mind network and the mirror neuron system network by a different group altogether. And they essentially found the same thing, that the two networks were correlated with one another, and that correlation was greater in more severe overall cases of autism, and it related to the degree of impairment on the ADI. So the general idea is that as you develop, networks should segregate. I think I have a summary slide. Yes, I do. Okay. Differential brain systems segregate with development under normal circumstance. There's less segregation of this important brain networks in ASD, and the degree of that segregation is related to the degree of social outcomes, so that the extent to which these networks are not well segregated and they're still cross-talking relates to poorer overall global problems with autism, which again, it supports this model of autism of having abnormal connectivity, abnormal functional connectivity, and most importantly, an abnormal developmental trajectory of connectivity. So we see this as things that develops over time, and as we look at the trajectory of development over time, we're not seeing that normal maturation process take place. And I'm just going to do one gene. The main point I wanted to make here was that we know that there are many, many risk genes that are involved in autism. Dan is going to be speaking after the break about the genetics of autism, and he will tell me how many genes there are. It changes every time he talks, so I don't know where we are right now, but we're at several hundred last time I heard him, which is just a few weeks ago. He's probably found 10 more since then. But you can imagine, with so much genetic variability, so many different genes contributing to this disorder, it makes sense that there's so much variability in the presentation of autism, because all of these genes don't all do the same thing. They all have their own separate jobs. They're going to affect the brains in different ways. And so then we would predict that the symptom pictures that we see in autism must be in some way related to the specific genetic background of that child. So that that child has certain genetic variants, we would expect to see certain kinds of variations in their brain that might be slightly different from the variations of a child with autism with a different set of risk genes, although obviously there's going to be some overlap as well. I'll tell you the bottom line is that we believe that the area of overlap is in how connections are made and formed in the brain. And how those connections are developed very, very early in life, and how they're formed and segregated, as I mentioned. That all of those autism risk genes, as Dan will point out, in some way relate to how neurons find one another and how they communicate with each other during development. But I'm going to talk about one gene that we know has a very specific effect, and that is the MET receptor tyrosine kinase gene. It's an ASD risk gene. I bring this one up. It is a gene that is involved in cell signaling and neural migration, so it's clearly important in finding connections and making connections in development. But one of the cool things about this gene is that it has a very specific expression pattern. It doesn't express all over the brain. It's expressed specifically in a few areas that we know are important in autism. The amygdala we've talked about, the hippocampus, the frontal cortex, a certain part of the frontal cortex, and also the temporal lobes, and in particular, the part of the temporal lobes that's important in face processing. So there are variations in this gene that have been associated with autism. And this is a study that was done by one of Marilla DePretta's graduate students, Jeff Rudy, looking at differences in brain activation in response to looking at faces, and faces bearing different emotional expressions, as a function of genetic status. So now we're going to separate the effects of autism from the effects of genetic risk. And in these pictures here on the left, we're showing differences that these are not differences in autism versus typically developing kids. These are just differences in genetic risk, regardless of diagnosis. And so what he found was, for those individuals who were in this, who had 2 copies of the risk gene, and these are people who had 2 copies of the non-risk gene, in response to faces, you can see, I think pretty much with your bare eye, that the activation patterns are somewhat different. And here is the difference between the 2 groups. And you can see that the risk genetic carriers have a lot more activation in those areas where that risk gene is expressed in the brain. And it's interesting because it suggests that how people are, how these face processing regions are responding is very much affected by a single gene that is expressed in this area of the brain. I always find it very shocking that one gene can have such a profound effect in the brain. And then you can also see the interaction by diagnosis. So this is in one of these big regions here in TD and in ASD. So in one of these face processing regions back here, we can see big differences in activation by diagnostic group, but we can also see big differences in activation just based on genetics. And this is the interaction between autism diagnosis and the gene. And you can see there's a very nice interaction here, so that if you've got the risk gene and you have autism, the activation patterns are significantly different. I think more to the point of functional connectivity, this gene affects functional connectivity when we put, didn't mean to do that. When we put a seed region here and we ask the question, how is the rest of the brain connected to this critical area of the brain? In the risk groups and the non-risk groups, what you can see is that the risk group, well, both have a certain amount of connection, but this is the difference between groups. There's much more connectivity across wide ranges of the brain in those individuals without the risk genes compared to those with the risk genes. So it suggests that these autism risk genes kind of lay a pattern of abnormal connectivity, of differences in connectivity. It's not enough to cause autism because we see it in typically developing kids as well as kids with autism, but it's enough to set a background of a certain kind of information processing, which combined with a lot of other factors may push the child over the edge into abnormal functioning in autism. So just to summarize overall, there is substantial variability in autism phenotypes and how every child with autism looks. You've seen one child with autism, you've seen one child with autism. These phenotypic variations, however, are associated with differences in brain structure and differences in brain function. And we think that it's very likely that it's the genetic risk factors, the different genetic vulnerabilities that underlie these brain behavioral relationships and these phenotypic variations. We're pretty much just at the beginning of trying to find all those pathways, but we think that's very important for really understanding the causes and the trajectories of development in autism. And we hope that if we understand these individual differences and how they're affected in the brain and how they affect behavior, that will ultimately lead us towards more individualized medicine, individualized treatment programs based on a particular person's set of brain differences and obviously ultimately their behavioral differences. And so with that, I will thank my close colleagues here, and I remind you to check out our website for any potential interest in studies. Thank you very much.